For the foreseeable future, the Wild Health Podcast will serve a very specific purpose. We have a team of over 40 MDs, PhDs, and expert specialists in emergency medicine, infectious disease, and other specialties who are constantly reviewing the emerging evidence and recommendations. Just to be clear, there's a lot of information out there. Everyone should follow the CDC and World Health Organization guidelines and recommendations. If it's easier to consume in an audio format, then listen in and we'll summarize and translate those recommendations and what we're finding. Every morning at 7 a.m. Eastern, we'll be releasing a podcast on a topic that we believe to be important to your health and well-being. Then every day at 7 p.m., we will release a breaking news update that will include the following: changes and new recommendations from the WHO or CDC, a message to our patients with updates on our protocols, supplement and medication recommendations, and testing updates as we increase capacity and expand testing, any other major updates that have happened or literature published that day, and we'll answer questions that we receive on Facebook or Instagram at Wild Health MD. With our network, we're in a unique position to help, and that's what we want to do. If you find this useful, then share with your friends and family and give us a 5-star written review on iTunes. That's the best way to get the information out to the largest number of individuals. Send us your questions as well, and we'll do our best to answer. Be kind to each other and be safe. Today we're here with Dr. Taylor Bright, our Director of Infectious Disease. Dr. Bright, we want to talk about some very specific things, some questions. I have some questions for you. I haven't talked to you today yet. I want to get some updates about our testing, and we have some questions actually from patients and other people through Instagram and Facebook as well, which I think you'll be an ideal person to answer these questions. We're going to jump right in. The first question I want to talk to you about is testing. I mentioned on our podcast recently that we, Wild Health, are working on doing everything we can to increase the supply of testing. I feel like this is a group effort among everyone. We have our big organizations. I feel very encouraged that they are working hard on this, but we still don't have adequate testing, so we're trying to do our part. And right now, your lab is trying to get a test out in 2 different ways. We're trying to hedge and do everything we can to make sure we have adequate testing. Can you tell me about the 2 different types of tests we're working on and any updates on timeline when we'll actually be testing with our test?
Yes, certainly. So currently on my lab, we are bringing on 2 tests, as you mentioned, Matt. One is the CDC test that is FDA approved for emergency use. And so this is a standard reverse transcriptase quantitative PCR assay, and this is the one that most labs are using. We're bringing this on. We received those test kits in yesterday, and we are currently running them in the lab. We're running control samples in the lab and looking to validate that assay so we can get that up and running. At the moment, we are on track to begin testing in the first week in April. I do want to say, though, that this situation is dynamic and that there are a number of clinical grade reagents that are in short supply as the demand, across the nation and across the world has skyrocketed. So we are doing our best to continue to stay on our timeline of providing coronavirus testing using the CDC test the first week in April. But it is a dynamic situation.
And that's a complex test, and you have the skill set, the equipment, both the lab equipment, the reagents and things, or we're working on getting them, and that's a great timeline you just gave there. Just to hedge our bets and speed things up even more, we invested heavily. We purchased a machine a few days ago that should give a 30-minute turnaround time for a yes or no SARS-CoV-2 test result. Tell me about the timeline for that. We're a little bit at the mercy of some shipping, but what do you know right now about that test?
Yes, so that test is what they call a sample to answer test. So this is a test that's going to be able to be run in a low complexity lab setting. So you won't need, you could run this even out of a medical clinic or even kind of some of these roadside testing that you hear kind of popping up around the country. So this is a very simple test. You're able to just drop the sample into the testing cartridge. It actually runs the same reaction in the cartridge that we run in our lab. It just does so on a smaller scale. So we're talking about doing one sample at a time in an asynchronous nature. It does look like it's going to provide results back in 30 to 40 minutes per sample, which is a great turnaround time. So we are bringing this on as our kind of rapid point of care diagnostic test. Because it is a sample to answer test, it has a little higher bar to go through with the FDA. The company that we are partnering with on this, it has applied for emergency use authorization from the FDA for this particular test. They're basing it on their influenza test, which they already have approved. So we are hoping that they will receive that approval and start shipping those cartridge kits again here in the second week of April. We're going to bring that machine on here hopefully early next week, and we'll be able to start doing basically influenza tests in the lab. This will help us kind of get the machine shaken down and get it ready to roll. Additionally, we'll be able to rule out those individuals who maybe have a respiratory infection to see if they do have flu. So that will be somewhat useful as we wait for the FDA to approve this low complexity sample to answer SARS-CoV-2 test.
Great. So those are the two tests we'll be able to run. The point of care test, we want that just because, that could lend itself to really great drive-through testing. Have that in the clinic. We don't need a clinical lab for that, so we want all options available. In the meantime, for our patients, we have swabs in. We can do testing immediately. We have the LabCorp test, which they're testing right now. We have those swabs. We've sent to some patients. We have other swabs in another clinical lab in West Virginia, which are testing. So we have 2 labs, actually 3 labs, that are testing right now. We're at the mercy of those labs and swabs, though, so we're trying to have our own testing also. So every avenue that we can find. There are multiple issues here, though. There's a swab shortage. We have LabCorp swabs. We have the swab for the test in West Virginia, but we have limited. We're supposed to get a very large number in tomorrow. This hopefully will solve those problems for us, the capacity issues. But again, we're supposed to get them in tomorrow. We don't have them in hand today. Hopefully we will by tomorrow. Taylor, why is there a swab shortage?
So the simple answer for the swab shortage is that the demand has increased 100x, 1,000x for these particular swabs. And the medical community just didn't have enough on hand. So the WHO suggested method for transporting these type of viral samples, this includes SARS-CoV-2 but also flu, is to use what they call universal transport media. And so that is what is normally used, but in this current global health pandemic, the resources have just been overwhelmed. The company that makes it is making as many as they can as fast as they can, but we just don't usually keep on enough inventory to cover 100x surge in demand over a 60 or 90 day period. So the companies are responding in kind. Additionally, the swabs themselves do have expiration date on them, so it's not like we can just buy a bunch, put them in the ground and wait for a pandemic. So this is just kind of one of those things. We saw the same swab shortage occur during the H1N1 pandemic in 2009. And so it's not unexpected, but it is a challenge that we have to overcome during the early part. Now, after the H1N1 pandemic, a number of labs recognized this problem and did some work to see what alternatives exist to the best-in-class universal transport media, or the UTM. And probably one of the greatest papers to go and reference and look at this is by Dr. Julian Druce, out of the Victoria Infectious Disease Unit in Australia. They actually took a number of different media, including universal transport media, viral transport media, some bacterial transport media, and even just pure saline, and showed that in a time of a shortage of swabs, specifically during a global pandemic, these alternative swabs can work just as well to be able to transport this media to testing facilities. Even they work just as well, even at room temperature for 4 to 5 days. So I encourage those individuals listening who are interested in this, check out this paper, use your professional discretion here, but there are alternatives as we wait for our industry partners to catch up and providing more swabs for us here.
Yeah, Taylor, we'll post that paper. If you don't want to email it to me, I know which one you're talking about. We'll post that on our Instagram for people if they're listening right now and they want to look at that paper. Because initially we wanted to use the UTC swabs. That is what the CDC recommends and is accepted, but there's other options. As you're saying, before I ask you a few other questions about swabs, tell me real quick what the capacity for our lab, timing, how many kind of tests per hour and per day we can probably run with both of the tests that we're trying to spin up.
So using the rapid diagnostic test that we're looking to spin up, we're able to do 4 samples at a time, about 30 minutes, so about eight samples an hour, and we can just keep running that thing for as many hours in the day. So that's about eight samples a day. So at that running at not full capacity, but let's call it running at a reasonable capacity, we can probably get 100 samples in that rapid diagnostic test. That particular test, again, because it is that asynchronous testing, i.e. you can test a person as soon as they come in. You don't have to wait for a 20 or 30 or 40 samples to come in to run at one time. Will have a unique clinical utility. So that's the rapid diagnostic test. For the CDC test, we're going to be able to run in that particular test 30 samples in a 1.5 hour run. So for that, again, we can just keep running that back to back. And additionally, we can bring on new equipment as we start to scale up. Each new machine we bring on will allow us to run an additional 30 to 40 samples per every hour and a half run. And if it really gets to it, we can actually really scale and go and run, find a different type of machine that allows us to run different plates, and therefore we can maybe get 90, 100 samples in that 1.5 hour run. So we're going to be able to do, hundreds of samples a day when we're at scale, and we'll hopefully provide a release valve for the testing demand as everyone chips in to kind of work to provide what we can.
And assuming we have all the equipment for that, or the swabs and things, and so the thought being, get drive-through testing, for example, swabbing a lot of people, we can run 30 at a time on the CDC test, and then the rapid test can run kind of 4 at a time, but run those quickly and asynchronously, get answers in 30 to 40 minutes. So it could potentially quite a bit of capacity there, probably 50 every hour and a half to 2 hour, kind of running both of those tests at the same time.
Yep, correct.
Let's talk about swabs real quick. You said that there are other swabs than the general swab, and we'll post the paper on that. Other than reading that paper, other people who are trying to do testing, any other recommendations? I know yesterday you mentioned that you could potentially make these swabs if you have the right equipment. Is that correct?
Correct. So you were able to make the swabs by making the media and filling tubes and sending that out. If you are a clinician in your lab and you have the swabs but not the transport media, the evidence that we are seeing is that you can drop that swab into a saline solution, just a normal saline solution that you'd have around, send that overnight. And again, it's not going to be if I was, in a non-pandemic situation, I'm doing my research, it would not be ideal. But in the current situation, we will be able to test that to see if there is a presence of SARS-CoV-2 virus. It will be more of a qualitative test instead of a quantitative test, but in this particular situation, we're just looking for a yes/no answer. So again, if you're out there and you don't have any of this transport media, or you can't get the test kits from either LabCorp or Copan itself, the guys who make the UTM, take that swab as you normally do, using a artificial tip and plastic shaft swab. And you can drop that in saline and ship that overnight, and that will be acceptable for the moment.
And this is not applicable to most people, but if you're a clinical lab and you have questions like that, reach out to us via Instagram. We can connect you with Dr. Bright and we can share that paper on how to do that exactly. I said UTC earlier, I meant UTM, so I want to correct that and make sure I'm using the right terminology. Questions that we've gotten that I think you would be great to answer just from listeners. A frequent question that we get, and we've had a group working on this, and it was not a very encouraging answer that you gave me, but I think we should talk about it. One question people frequently have is, once we get this, because we are looking at a very high percentage of infectivity rate with the R0 that exists with this virus, are people immune? Can you get this again after you've been infected once?
So having reviewed all the available evidence, and obviously the available evidence increases daily, my professional opinion is that there will not be a strong neutralizing immune response after being infected with SARS-CoV-2, i.e. there is the possibility of being reinfected by this virus at some point in the future. So that is not necessarily the most positive answer, so I want to take a minute just to kind of walk through what we're seeing and walk through the trajectory of data that we have on this. So the a priori probability that we would be able to mount a effective immune response to a coronavirus based on what we know about the coronavirus strains that cause the common cold, the coronavirus strain that caused SARS in 2003, and the coronavirus strain that led to the MERS outbreak in 2012. Based on our knowledge of those viruses, we would not expect a human being to mount a strong neutralizing antibody response to a coronavirus. You can just think of maybe you get 2 to 3 colds every cold and flu season, year after year after year. So there's a sense that we are not able to mount strong immune responses to this family of viruses. So then once we got into the outbreak, right around end of February, there were reports that people were starting to show signs of relapse infection, whether this was what they call a recrudescent infection, i.e. the viral load decreased in your body to an undetectable level and then increased, or you actually cleared the initial virus and then were reinfected, is unknown. But irrespective of which of those was the cause of the relapse, it again argues that a strong neutralizing immune response is unlikely. So recently, we've also seen some reports showing experiments in rhesus macaques that these monkeys did mount a neutralizing immune response, i.e. they were not reinfected after re-challenge with the virus. They were re-challenged 28 days post-infection. So that would argue that there is a short-term immunity, which would be good. But this study was very small. It was only 2 monkeys that were re-challenged, and one of which was only followed for 5 days. So encouraging data that there might be short-term immunity. And if there is short-term immunity, maybe there's long-term immunity. But at the moment, we do not have a lot of strong data that you will be immune after infection with this virus. Now, the best data for this is going to come from the ongoing vaccine trials. Those have started. There's an ongoing phase 1 safety trial in Seattle. And these are looking at vaccines directly related to the viral protein that leads to infection of the human cells. So we'll get really good data of the human immune response after these trials, and that will really be the gold standard in helping answer this question.
So we're getting more data, but in general, just to summarize, it's not clear that we're going to be immune from this after we're infected. How would you compare this to kind of cold, flu, other viruses like that in our immunity afterwards?
So what it looks like,
or even things like chickenpox, which we get once and then we don't get again.
Yeah. So what we're looking at, the opposite ends of the spectrums here that would probably be most resonant with the audience would be chickenpox. You get it once, and then shingles later in life, notwithstanding, you are immune from reinfection with varicella virus. The other end of the spectrum is the common cold, when again you may have 2 or 3 common colds throughout a cold and flu season. It is more likely, since common cold is caused by a coronavirus, and this is a coronavirus, that we will be on the side of the immunity spectrum that is closer to common cold, influenza, where you may be able to get reinfected with a strain, maybe not immediately, maybe not 2 or 3 months, but maybe season to season. So now that being said, there will be people who genetically, because of their own genetics, will mount strong immune responses, and there will be people that mount weaker immune responses. So what we really need to see is how does the population as a whole mount an immune response to this virus. So then we have a good sense of herd immunity, and then we have even a better sense of how our non-pharmaceutical control, so that's the social distancing, travel restrictions and that type of thing, to kind of see how those are going to play out with regards to increasing our herd immunity.
You just said some people will mount a response better or worse than others. Anything we can do to affect that as individuals?
Not that I'm aware of.
Okay, I assume that's what you would say. You and Mike and I were discussing a paper which is ancient history at this point because it's almost 3 days old, but published in Nature. It's titled "The Breadth of Concomitant Immune Responses Prior to Patient Recovery: A Case Report of Non-Severe COVID-19." What does this paper tell you about immunity, and then also the promise of a vaccine?
So this paper followed an individual who was infected in Hubei province and then traveled to Australia. This individual was identified as being infected with SARS-CoV-2 and was tracked very closely. They did a lot of great immunology, and they were did a great job tracking the natural history of the immunological response to this particular virus in this one patient. So again, this is one patient's response. But what we did see was we saw in this particular patient, there was a robust immune response. That's a acute immune response that we would activate towards any viral infection that was able, in this case, to clear the disease with only moderate symptoms. This individual ceased to be SARS-CoV-2 positive. That's via the testing at day 7, and then continued to make a full recovery after only a moderate disease in this particular situation. So what we've seen here is a very good understanding of how the body's acute response, predominantly, let's call it a CD8-mediated viral response. This paper tells us relatively little about what the long-term immunity of this individual or any individual would be. Basically, it shows that we can clear the virus, and then this person will have to be followed for longer to see if, 1, they are reinfected, or 2, they're able to show long-term presence of neutralizing antibodies in their bloodstream, which would argue for immunity in this particular individual.
Great. So, and I apologize if some of this is not relevant to everyone, but we're trying to speak both to physicians, clinicians, and a general audience at the same time. So I'm going to jump to just kind of some general questions that people have sent. I'm pulling these off of our Instagram. It's at Wild Health MD right now. So be sure and send these in. We're going to be answering these Instagram and Facebook questions daily in the evening and sending out an update. So keep sending these. One question is, what does it mean when it's called novel?
So in this particular use of the word novel, it can be synonymous with new. So this is a coronavirus strain that is new to the human race. We believe that it is very similar to a bat-like coronavirus. So we believe it jumped over from bats, i.e. it's what's called a zoonotic infection. But in this case, novel just means that it is a new strain of coronavirus that we as a species have never seen.
Another question is, can you have the flu and COVID at the same time? I saw a study, and you can add to this, but so what we're asking is, what is the co-infection rate of a different virus and SARS-CoV-2 at the same time? And there was a Stanford study that just came out showing that as high as a 22.6% co-infection rate, which matters because a lot of times we think, well, part of our protocols right now are we test for, because there is a shortage of the test, we test for other viruses first, like we send a flu swab, and if someone is positive, then we assume, okay, that's what's wrong with them. That's why they have a fever. But this study, I guess, casts a little bit of doubt on that and says, no, you know what, you can definitely have this novel coronavirus as well. Any other data that you know related to co-infection rates?
Not that I'm aware of. That particular study that you cited is the current gold standard with regard to co-infection.
Another question on Instagram. Someone is interested in home interventions and recommendations for how best to address and prevent the IL-6 surge, i.e. the cytokine storm. They heard that steroids are associated with worse outcomes. And so I'll go ahead and say we did address this on a prior podcast. It seems like early steroids actually may increase shedding and contagiousness. So it's probably not a good idea initially. And maybe there is some benefit in the hospital. But if you're home, probably not steroids initially. There's also some negative data on ibuprofen and NSAIDs here, but that's still, we're gathering more data about that. What they specifically ask about is, what about PRN, quote-unquote, rescue inhalers and nebulizers with the reactive airway disease brought on by a hyped immune response? Okay, so we'll cover that question a little more later. We'll dig into that. We don't want to just give you an off-the-cuff answer. We'll do some research on that. Again, send more questions to the Instagram and Facebook page, and we'll try to answer them both there and on the podcast. What we're going to do going forward is every morning at 7 a.m. release a podcast on things that we think are important, and then at 7 p.m. release an update, which will be, okay, what are the current CDC and World Health Organization recommendations, anything new that's going on, what breaking news are there, and we'll answer your questions as well. And then also we'll probably speak directly to our patients because our protocols are changing daily with what we find out to be true, and that's going to change all the time. So we're occasionally going to be wrong. So again, I would encourage people to follow these CDC and WHO guidelines as much as they can, and we'll try to be very up to date as well. Anything else that you think is really important to talk about right now, Taylor?
I think as Matt said, listen to the CDC, WHO guidelines, tune in as much as you can. Continue to keep yourself informed. This changes every day. The situation is the word dynamic does not quite cover it. And then other than that, take care of yourself and each other. That's the way we're going to get through this.
Yeah, and I actually just got a couple questions that just popped up on Instagram. So one was they've seen some things about hydroxychloroquine, potentially prophylactically and for treatment. So we talked about hydroxychloroquine on a prior podcast and using that with zinc. We will give you an update on that. We actually have a pharmacologist looking at this question. We have kind of a protocol for our patients already, but we're going to dig in a little more before we talk about that on the podcast. Another question just popped up is, yesterday we released a podcast and we talk about just general supplements and nutrition in general. And we talk about making sure your vitamin D and vitamin A levels are adequate for immunity. There's been some talk on some podcasts and other places about not taking extra vitamin D and vitamin A, and that potentially causing some issues. I know what this person is asking about. I heard that podcast, and I do not think there's any evidence for not taking those. I don't want to dig too much into this answer, but someone asked this on Facebook. We'll post an answer to that, but I do not think you should change anything you're doing right now with regards to vitamin D and vitamin A. I don't think there's any evidence for that. Otherwise, keep putting those questions in. We'll keep answering them. And thank you for joining us, Dr. Bright.
Thanks, Matt.
Thanks so much for listening to this COVID-19 update. Please send us your questions or follow along on Instagram and Facebook at WildHealthMD. And just to be clear, this is not medical advice. No patient-physician relationship has been established, and this resource is really for general informational purposes only. And finally, if you want to spread the word, please send this podcast to friends and family and give us a review on iTunes. Thanks again, and stay safe.